I'm going to be talking about some work from this paper here, which was also a lot of what I worked on through my PhD. It's finally published. I'll have a QR code on the last slide as well. I'm currently a postdoc at the University of Maryland. Um, and yeah, the paper link also has our associated GitHub, which if you'd like to run or repurpose any of the models shown here, that's what it's there for. So yeah, I'm just going to talk about some genomics-informed agent-based models of the PDAC tumor microenvironment. So I'll start with an overview of the pancreatic cancer microenvironment, and then talk about the relationship between like cancer-associated fibroblasts and the epithelial to mesenchymal transition in tumor cells. Um, and then talk about some experiments we did and how we turned them into an agent-based model. And then I'll show two genomics and agent-based model integrations, one forecasting tumor growth from spatial transcriptomics initialized ABMs, and then one from single-cell RNA sequencing data set. So first, the PDAC microenvironment. Pancreatic ductal adenocarcinoma is the most common subtype of pancreatic cancer with a 5-year survival rate of only 13%. A hallmark of PDAC is what is called a desmoplastic tumor microenvironment. Compared to the healthy tissue on the left, PDAC on the right has masses of cell growth in places that used to be healthy ducts and is infiltrated by a greater diversity of immune and stromal cells while being crisscrossed and encapsulated in this network of collagen fibers, which are deposited by cancer-associated fibroblasts, or CAFs. I'm going to refer to them interchangeably throughout this talk. I'm so sorry. Um, so this dense fibrotic network of collagen is referred to as desmoplasia and is seen in certain types of tumors. It provides a physical barrier to the tumor mass and is thought to be also a barrier to like therapies physically making it to the tumor. The immune cells that are found in this microenvironment are often they're there, but they're often sort of in immunosuppressive or a tumor protective mode, and we'll discuss that a little bit later. The tumor microenvironment is comprised of these interacting cell types which work together to enable and support tumor growth and metastasis. And this desmoplasia really develops alongside the tumor as the dense extracellular matrix is deposited and remodeled by the cancer-associated fibroblasts. The changing environment itself is thought to support the tumor's growth, progression, and ability to metastasize. The pancreatic tumor cells themselves, like even at like late stage tumors, are relatively few, comprising less than half of most advanced tumors by like cell counts. So we wanted to start modeling these processes and tease apart like what cells in the microenvironment are supporting this in what ways. So first we wanted to look at the specific role of fibroblasts in inducing the epithelial to mesenchymal transition. So when we looked at a pre-treatment single-cell RNA sequencing data set at the different tumor cell phenotypes, this epithelial to mesenchymal, sorry, transition and inflammatory pattern of gene expression that we identified in epithelial cells correlated strongly with the presence of fibroblasts per sample. So the tissues with the most cancer-associated fibroblasts also had the highest amount of expression of this gene signature, which indicated inflammatory response and epithelial to mesenchymal transition. Follow-up experiments in our organoid co-culture system confirmed that this gene set is induced on average when these pancreatic epithelial tumor cells are grown with fibroblasts. So we now have a model like this where we have epithelial-like tumor cells, which this is sort of our lab's own classification of like the different phenotypes of PDAC tumor cells that can exist, but we have this epithelial-like, which is mainly in a proliferative state. So it's dividing, but it's probably not particularly inflammatory and it's not moving. But then when they go into this mesenchymal-like state, they become able to be motile and more inflammatory, we think. So we wanted to know, we think that the epithelial to mesenchymal transition is supported by the fibroblasts, either through cell contact or through a substrate that they secrete. Whereas the mesenchymal to epithelial transition is sort of what happens like quote unquote like naturally when they're not under like another influence. Um, so we thought a big part of this that we could sort of measure and model is like the the aspect of becoming a mesenchymal cell that is like becoming motile. So we turned to our collaborators in the Philip lab at Johns Hopkins, and we performed a single cell tracking experiment using pancreatic neoplastic and then uh, fibroblast CAF cell lines. So we quantified the cell velocity at varying collagen concentrations in like cell line monoculture and then co-culture. Um, so we found that the two cell lines, the fibroblast and the tumor cell, have different responses to the ECM density changing. So the ECM density changing here is essentially like the we made like matrigels with different concentrations of like collagen 1, which is like our best approximation of what extracellular matrix is like. Um, and then using this tracking microscope, which has like an algorithm that refocuses and regularly images moving live cells that the Philip lab has developed. It's like very amazing. Then we were able to get all kinds of information about how like about the motion of those cells. So not only like the instantaneous velocity, but also like how much turning are they doing and like the progressivity of movement. And it's very crazy. So this is some of the information that we got out of this. So we found that the tumor cells, so like the panc cells here, have varying motility depending on the concentration of ECM, like by themselves. With the peak motility being two peaks, one at 4 mg per mil and then the other at 7. Um, However, when they were with the fibroblasts, they were just essentially faster across the board by like these sorry, my p-value got cut off here, but this is significant. They're faster across the board, and then also they peak at 4 mg per mil and then not again when they're in co-culture. So we saw that tumor cells get a motility boost at certain concentrations of ECM when they're co-cultured with CAPS. So then given these relationships, I wanted to represent these cell processes in a computational agent-based model. So agent-based models are computational simulations of interacting agents. I wanted to simulate tumor cell behavior to forecast tumor growth as a whole and development. So the idea is that observations about cell behavior can become model configurations and rules. And then those can like that can become like the model configuration that for the thing that you can simulate. So agent-based models comprise populations of interacting agents, and each agent entity acts at each time step according to its internal state and behavior rules. So the next state that it experiences is going to be determined by like its previous state, its actions, um, its the other agents, and then like its environment. Um, and the agent-based modeling like the framework, um, recomputes that at every time step, essentially. So it is like it's a stochastic simulation. It's um, it's just going down one path at a time of what interactions could possibly happen. It's not trying to create like a tree of every possible interaction, if that kind of makes sense. So it's like it's sort of it is a fairly, to me, what feels like a relatively computationally efficient way of like simulating like possible cell behavior. Um and because the runtime basically scales with the number of agents that you put in and the frequency of the time step, of course. Um, so all of the models that I'm going to show here are in PhysiCell, which is an agent-based modeling framework that is made for cellular ABMs and is built by like our collaborators, the Macklin Lab at Indiana University. So, for instance, this is a prototype model with these cell players. So the gray cells here are what I call like like normal epithelial cells. So they don't have tumor cell-like behavior, but then these blue and these green do. The green are the proliferating type and the blue are the motile type. Um, this is before we did the experiment, so these are just the guess motility values we put in. And you can also see these yellow agents here. The fibroblasts are depositing this substrate, which is extracellular matrix. Um, and we can see here that the cells, like, they become motile, they move away from the tumor mass, and then some of them like have switched back into proliferating type, and um they're forming, like, I guess what I would call like these satellite colonies off to the side. So then to get the right cell motility behaviors, because I kind of looked at that and I was like, um those cells are, I don't know if those cells are moving how they're meant to be moving. So that was actually the point at which we did the experiment. Um, so the measurements from earlier can be converted into like behavior response rules in PhysiCell. PhysiCell recently added this, um, they call it a rules grammar, but what it is like that you can specify um, how a behavior changes in response to a signal just using like this human readable grammar. And what it does is it parses that and it converts it into a Hill function. So it makes it really easy to program in things like, for me, I really like to model, you know, in my free time aside from this, I really like to model T cells. So um, you can say something like T cells, the proliferation goes up when they see more IL-2. And that can be like, it can be a switch function. It can be like, Hill functions can approximate a lot of the biological response curves that we want to approximate in models, if that makes sense. So anyway, with two of these Hill functions, which here are expressed in like in words, um, and okay, we were able to fit the um, measured motility data using a combination of two Hill functions. In this case, it was just a strange curve to fit. For whatever reason, it was one of the situations where a Hill function does not very well approximate it. But anyway, two of them completely did the trick. Um, so this experiment gave us accurate like base and max motility values for both of these cell lines. Um, and then it also gives us the relationship between motility and substrate density. So now the agents should move approximately the right amount per simulated time step, and the cell types velocities are calibrated accordingly with each other. Um, so simulating, now this is like a simulation of um a mixed um so pancreatic tumor cell, and then fibroblasts, like a mixed spheroid, which is like an experiment we do in the lab. And so I made like simulations of um those spheroids at growing at different cell ratios of like CAFs to tumor cells, and then we were doing this in the lab at the same time. And now that we have the correct motility behavior, I'm seeing like invasive growth, but like it's looking a lot more similar to what we see in the lab, which is that the um invasive parts, I guess what you could call it, which um would be um uh these fronts of like proliferating type green cells, which it's a little hard to see here, but that's actually what's shown here too. The invading part of this spheroid is um, all green, no magenta, so there's no fibroblasts in this invading part. And this is also, the green dye is a proliferation dye, so we also were able to tell that these cells had divided at least once that were in the invasion area, which may like just it may seem kind of common sense, but like when it actually comes down to proving like whether a tumor cell moved somewhere or grew somewhere, it's not immediately obvious always. um so yeah, so now I was like, I had this matched simulations with the experiment. And so once I had the simulation at each um, cell type ratio, um, calculated the invasiveness of each spheroid. And so like the model here is able to make like a fairly clear prediction about which ratios of cells it thinks will produce the most invasive spheroids. Um, which then the intent was to compare to the invasiveness metric of our spheroids that we grew in the lab. However, we had like an imaging registration issue with that data, and so we actually ended up sort of having to make a whole image correcting thing tool to even work with that data, and that's actually in press now at Cell Systems, Kramer and Lopez Vidal et al. Um, and that data is going to be there. So um, yes. So the model can now make clear predictions as to which ratios should induce the highest degree of invasive growth. Um, and now I wanted to use these rules of cell behavior that had worked for the spheroids to forecast tumor growth from genomics-based initial conditions. So while the spheroid and cell tracking experiments allow us to interrogate behaviors of cell lines, spatial transcriptomics data is able to capture specific spatial arrangements of cell neighborhoods, in addition to pure abundances, and in actual human tumors, um, which have the real diversity of human tumors and have been developing for as long as human tumors have. That is like an obsession that I have, is that human genomics data gives us like all the information we need about, like, maybe not all of it, but it gives us like information that we don't, it's very hard to get otherwise about human immunology. Um, so I thought I could use spatial transcriptomics data to get initial states and positions for like a cell model. And then if I use my existing model rules, it would be able to like predict what would happen like per tissue and like in each area of a tissue. So the idea, um, and now this is a tool that you can use, but the idea is that processed spatial transcriptomics data contains positions and annotated identities for each spot profiled. So this is like not single cell resolution, but it's like single spot resolution. Xenium data import, which I'm now doing, is much better at giving you single cell. This information should be able to be used to specify like cell types and arrangements in an agent-based model too. So Daniel Bergman in our group developed the tool that like lets you do this like point and click. But how I did this for the first time, before we had the tool to automate it, to capture all of the agent types that I wanted to represent, I used a combination of like transcriptomic annotations of the spot. So like the gene signatures that I talked about way earlier for the epithelial and the mesenchymal-like cells. And then also I was able to use the matched H&E image that comes with every Visium data set to and score it with there are these like AI image annotation tools. There's this one called Coda that we used by Dr. Ashley Keeman that's like essentially trains on pathologist annotations of like existing annotated tissues and then can annotate a new slide that you give it. And this is like a pixel level annotation. And one thing it's really good at is telling you where there's collagen fibers in the image. So I used that detection to say where should the collagen be densest at the beginning of the simulation to provide like structure to the tissue. And then most everything else is scored from transcriptomics. And so now from 2 tissue slices, um, uh, I have 2 initialized agent-based models, and then when I play the simulation forward, um, we can see like how, what's predicted in each tissue. And here, like, I mean, I basically just see pockets of proliferating cells like arising in areas that were low density in the tissue earlier in the simulation. So another way to use this, like if you were using this with your data, type thing is to or in addition to changing the initial condition, like what tissue you put in or how the cells are arranged, you could keep that the same and instead change the mechanistic assumptions that go into your model. So like, what are the response rules or like what are the cell behaviors that you're assuming? So here is an example of that. So this is from one initial condition tissue. And then this top um, movie here is saying, what if we assume that the epithelial to mesenchymal transition is happening when in response to the ECM substrate? So is ECM inducing and supporting this transition? Or on the bottom, is it just cell contact with the fibroblasts inducing this transition? And then ECM, it's still there, but it's just modulating migration. It has effect on motility, but it's not actually the thing that like keeps cells in the mesenchymal-like state. So I think what this let me at least see is that the difference in those 2 assumptions of how it works is that if it is a substrate-driven mechanism, then the areas of mesenchymal-like, so the blue, separating the proliferating pockets from like the surrounding tissue and the fibroblasts, is much broader because like ECM has been deposited there, and so there's like a wider area where like they'll be kept in that state, I think. Whereas with the cell contact based mechanism, that transitional zone, the blue, is only like 1 cell thick because the only thing keeping them in that state is the contact with the fibroblasts. So when this assumption is in place, then you see these much bigger proliferating pockets of these green cells than you do here. Like here you see some, but they're not like pushing out like and this large. So anyway, just I think that those are sort of right now the two main ways I can think to use Physi-cell spatial transcriptomics. Either you have an idea of how you think the system should work, and then you simulate from a cohort of tissues. Potentially, if you have matched different like lesion stages, I think it would be a really cool project to see like if you can or cannot forecast between them. Um, and then the other way being to vary the mechanism and say, if I just change this small part of my hypothesis, what does that imply about the system? So just the last experiment we did here to follow up on that. So we felt pretty confident that the fibroblasts are inducing EMT and invasive behavior in our model in a way that recapitulates like pretty well what we see experimentally. So we wanted to try to delineate between those two possibilities here, the cell contact based or the substrate based EMT. Um, so we performed an experiment using patient-derived pancreatic organoids, which showed us that CAF condition, so fibroblast condition media, was sufficient to induce invasive growth, um, which um, that doesn't rule out a role for cell contact in inducing EMT, but it does rule in a role for substrate based, which to me, like whatever substrate the calves secreted into the conditioned media is probably something like what they're secreting and providing like in addition to the ECM and like providing to the tumors is the idea anyway. So we found that whether you use the myofibroblastic CAFs or the inflammatory CAFs, MyCAFs or iCAFs, you got an increase in this in the mean inverse circularity, which is like essentially just a measure of how not circular the spheroid is, because they all start out spherical by nature of the assay, and then the more invasive ones will become stranger in shape. So you can use this basically tracing of the perimeter to get like a measure of invasiveness, and that's kind of the main way people do it, it seemed. And yeah, for this assay, we worked with Dr. Laura Wood and Fatima Shojin at Johns Hopkins, and they were amazing, of course. So now that I had a method to forecast tumor growth, I also wanted to try and forecast immunotherapy response. So immune checkpoint therapy or immune checkpoint inhibition, ICI, has been the cancer treatment revolution of the 21st century, with the revelation that the body holds the capacity to destroy most tumors if the brakes are taken off the immune system in the right way. So put another way, almost everyone has T cells specific for PDAC tumor antigens. Most people have them in the PDAC tumor microenvironment, but they are being actively stopped from killing the tumor by the tumor, and also by its microenvironment, by the stroma, by myeloid cells, by other T cells, by Tregs. Essentially, like uh, it's not that the fighters aren't there, but they're just locked in. Yeah, I can't think of a word for it, but so PD-1 and PD-L1 is a classic example of an immune checkpoint that has been successfully exploited as a therapy target. and that's shown here. So PD1 on a T cell it's called programmed death ligand 1. That's a little dramatic. Uh, Engaging it can, in theory, like induce the T cell to die or like become anergic. And that's like quote unquote why like honestly, a lot of tumors express PDL1, which is its ligand. Um, but also a lot of T cells, once they've been activated, they also express PD1. So it's not always like just a sign that something bad is going to happen to the T cell, but that engagement alongside TCR signaling is something that can like shut off a T cell basically. So when that tumors don't eat me signal to the T cell is blocked by like a blocking antibody or a small molecule inhibitor, whatever it is, the tumor killing can commence. However, immune checkpoint therapy so far has had limited efficacy in PDAC. So that's thought to be due to the fact that PDAC has a very immune cold microenvironment. So that's different from like uh, not having the immune system there. Importantly, it's a common perception that like there's just no immune cells in PDAC. But that's not true. I would like to evangelize to the world. Um, so where this is where most of the myeloid cells are suppressor cells or pro-tumor macrophages called TAMs that sort of work to keep the tumor healthy and shut down any cytotoxic T cell that comes too close. Regulatory T cells or Tregs also serve this purpose and actively prevent other T cells from maintaining activated status in the tumor vicinity. And a typical immunotherapy responsive tumor that we would think of by contrast has antigen presenting cells. So dendritic cells, this could also be macrophages, but dendritic cells are the kind of gold star at this, that will activate T cells. And the infiltrating T cells will not be exhausted. They'll be in an effector state. PDAC is immune cold, so immune targeting therapy strategies, such as the one I'm about to talk about, is typically trying to target multiple players at once, to like hit the immunosuppression at multiple points. So um, you're not just turning on antigen presentation, you're also turning off T cell exhaustion. Like because most of them alone don't seem to really work. Um, like, so specifically the trial that I wish to create on um, the genomics data cohort that I had used a neoantigen tumor vaccine um, called GVAX, which targets dendritic cells. In my model, there are no dendritic cells, so when I modeled GVAX just as an expansion of T cell populations, which in theory is because of the dendritic cells. Um, so with or without added PD-1 checkpoint therapy, our genomics results also suggested that modulating CD137 with the agonist urelumab could additionally activate T cells. So I wanted to consolidate our PDAC untreated single-cell RNA sequencing data set into my approximated phenotypes of interest for my model. Um, and so the first step was to translate the single-cell metadata into a set of per tissue counts for each population of interest. I did this using graph-based clustering annotations here, plus looking at the gene transcripts for PD1, PDL1, CD137, essentially trying to type each cell by its expression of therapy target genes. And then applying each therapy, which is a reassignment of cell phenotypes alone in combination to each tissue. So in my model, um, what nivolumab treatment is a reassignment of all of the PD-1 high cells to their PD-1 low counterparts. So all of the PD-1 high, CD137 high, CD8 T cells, for instance, now become PD-1 low, CD137 high, because their PD-1, even if it's there, it's now blocked. So I consider the PD-1 low. What this means is that that T cell now can kill PD-L1 high tumor cells in addition to PD-L1 low because the blocking mechanism is not there. And then a similar logic for the anti-CD137, except for now this is an agonist antibody. So instead of shutting it off, it should like turn it on. So either the populations that are CD137 low now become their CD137 high counterparts. They now secrete inflammatory factor, and they now kill at like a slightly faster rate as well. As I mentioned before, the PD-1 transformation also happens with the CD4 T cells. And as I mentioned before, the GVAX is just an expansion of T cells across the board. So this is showing baseline and then 3 monotherapy simulations for one tissue. So this is like for one, demultiplex tissue in the single cell RNA sequencing data set, if I take the T cell counts and proportions and then like the tumor proportions and the number of macrophages, and I set up this like simulated like co-culture experiment, then I have like this approximation of like what my model predicts could happen in this microenvironment. Then I can say, let me expand the T cells, let me see what happens. Let me make the T cells PD-1 low, see what happens, and then make them CD137 high and see what happens. So this is, so this was kind of like my proof of concept for this model. And I saw that none of these alone effectively eliminated this simulated tumor. So then these are the double and triple combination therapy simulations for that same tissue. So this is each 2 paired together and then all 3. If we assume we can influence the correct targets, this predicts better immune response with the triple combination therapy in this simulated patient. So then the idea with this system is that you can run each therapy condition for each tissue, and then you're able to get like a prediction of how the model thinks each tissue will respond to each therapy under our phenotype classification and behavior rule assumptions. So the most effective therapy, and whether any therapy was effective at all, was varied greatly by patient. Here in tissue 1 and tissue 12, we can see the triple combination is like clearly the most effective, and no other reached that threshold. However, here in tissue 4, it even just urelumab can essentially get you to that same level. So there were other tissues where the triple combination like it did worse than so it was you get like a ranking of the therapies per tissue. So when we tried to find the most important factor determining whether a simulated tumor grew or shrank under therapy compared to baseline, the only cell population that showed a significant difference in responders versus non-responders, simulated responders versus non-responders, to the triple combination therapy was macrophages, whereas CD4 and CD8 T cell counts at baseline were not significant in terms of predicting like therapy response, which surprised me because I really thought that just the more that this model doesn't have that many things going on in it. And to me, it just seemed that the more killing cells you have, the more killing is going to happen. But actually, it mattered far more how many macrophages were mixed in with that tumor mass in the center at baseline. And just an interesting note that later like signaling analysis in this clinical trial that actually ended up happening showed that these TREM2 positive like immunosuppressive macrophages went up in that triple combination, um, and that they were signaling to fibroblasts and tumor cells. So just sort of just an interesting note and something that was in concordance with um, the model. So we just were when we got these results, it was something to look back and compare. So um that is essentially everything I have. So I'll just say the work I've shown today has focused around how to connect omics and agent-based models to understand the PDAC microenvironment and how to build personalized tumor forecasts that can be individualized and reevaluated. We can use clinical data to decipher cellular states and turn those cell states into a forecasting model. So using computational methods to explore certain cell community processes that protect the tumor and allow it to progress will lead us to new interception strategies for this devastating disease.
Thank you.
My
Yes, so sorry.
my summary and takeaways are just that using these kind of computational models can help us understand emergent cell behavior and forecast outcomes in the tumor microenvironment with unseen therapy and combination therapy regimens. Genomics metadata, I think, is a very good candidate to translate directly into model initial conditions. And ABMs are a good way to explore your hypotheses in a genomics data set. And then clinical genomics data and computational modeling can give us direct insight into human tumor immunology. So I'd just like to acknowledge everyone who contributed to this work, especially my mentor, Dr. Ilana Fertig, and say just thank you again for the invitation.